Antibiotic resistance is one of the greatest medical challenges of our time. Bacteria evolve rapidly, developing mechanisms to survive even the strongest drugs. While random mutations can cause resistance, many microbes rely on genetic exchange, sharing genes that encode resistance enzymes, efflux pumps, or target modifications. These exchanges occur through plasmids, transposons, and a powerful system called the Integron, a genetic mechanism that collects and expresses resistance genes. Understanding integrons helps explain how bacteria adapt faster than medicine can respond. To understand this process, let's look at what an integron actually is. An integron is a specialized DNA region that captures and expresses genes. It has three essential parts: the integrase gene, which encodes an enzyme for recombination, the recombination site, where new DNA segments are inserted, and a promoter, which drives gene expression. Together, these components form a genetic framework that acts like a library, allowing bacteria to store multiple gene cassettes, each potentially carrying a new resistance trait. But how does an integron actually capture and use these genes?
When a bacterium encounters a free gene cassette, the integrase enzyme recognizes a recombination site called ATTC on the cassette and joins it to the integron's ATTI site. This inserts the cassette into the DNA sequence, forming a growing array of genes. The promoter at the start ensures that genes nearest to it are most actively expressed. Under antibiotic stress, the integrase can rearrange these cassettes, moving vital resistance genes closer to the promoter, a built-in genetic adaptation system that operates in real time. Integrons vary among bacteria, and some have evolved into entire classes with different roles. Integrons are divided into several classes based on their integrase gene sequences. Class 1 integrons are the most common in clinical bacteria like Pseudomonas aeruginosa, Klebsiella pneumoniae, and Escherichia coli. Class 2 and 3 integrons appear less frequently, but play roles in environmental or commensal bacteria. Beyond hospitals, countless environmental integrons exist in soil and water microbes, forming reservoirs of potential resistance genes. These environmental integrons can transfer into pathogens, linking ecology to global health. Let's see a real example that shows just how powerful these systems can be.
In the aquatic bacterium Vibrio cholerae, scientists discovered a superintegron containing hundreds of gene cassettes. These genes encode diverse functions: metabolism, signaling, and stress responses, many unrelated to antibiotics. The superintegron acts as an evolutionary archive, offering Vibrio the ability to adapt to salinity, temperature, and nutrient change. This discovery proved that integrons originated as tools of environmental flexibility long before humans created antibiotics. Now, let's move from the ocean to the clinic for a second example. In hospitals, Pseudomonas aeruginosa carries class 1 integrons packed with resistance genes. Many encode enzymes such as VM-type beta-lactamases, which destroy carbapenem antibiotics. These integrons often sit on plasmids or transposons, moving easily between bacteria. During outbreaks, sequencing reveals nearly identical integron structures in unrelated strains, proof of rapid horizontal gene transfer. This mobility makes class 1 integrons a driving force behind multi-drug resistance in intensive care units worldwide. Finally, let's look toward the strategies being developed to control these systems.
Modern research targets the integron itself. Scientists are designing integrase inhibitors to block cassette recombination and exploring CRISPR-based tools to delete integron regions from bacterial DNA. Surveillance programs now monitor inti genes as markers of resistance spread in hospitals and wastewater. Yet, the most effective defense remains antibiotic stewardship, using drugs wisely to reduce the pressure that activates these systems. Understanding integrons offers not just insight into bacterial evolution, but a roadmap for preserving the power of antibiotics. Integrons remind us that even the smallest genetic elements can shape the future of medicine. They connect the environment, evolution, and human health in one continuous network of adaptation. Every discovery about how these systems work brings us closer to new ways of preventing resistance before it begins. The challenge is immense, but so is the opportunity to use our understanding of microbial genetics not just to fight disease, but to stay one step ahead of evolution itself. Knowledge is the most powerful antibiotic. Integrons, the hidden engines of antibiotic resistance.